good morning. As was already said, my name is Hlubinová, and today I would like to present joint work with my PhD supervisor Pavel Bokes and Professor Singh on a matched asymptotic analysis of the Luria-Delbrück distribution in a reversible fluctuation assay. So in this short presentation, I will start with a brief motivation, biological motivation behind our research. Then I will move on the mathematical formulation of the model, and I will sum up with some results we obtained. So, do mutations in bacteria arise randomly, or are they induced by the environment? This fundamental evolutionary question was asked by Max Delbrück and Salvatore Luria in 1950s, and the answer came through what is now known as the fluctuation test. So, what it is about? In their experiment, Max Delbrück and Salvatore Luria, they had many identical copies of a bacterial culture in parallel, and they let them grow. After some period of growth, they exposed each culture to bacteriophage T1. And since it is bacteriophage, only those bacteria that are resistant may survive, and they grow into colonies, and all others become extinct. So, and therefore, there are two possible ways. What can they see? At the left, we can see that there are roughly the same number of cultures in each agar plate. That would mean that the Lamarckian theory is right, saying that the mutation only is present only after the exposure to the virus. However, that was not what they saw. Their plates contained much variance. The number was contained much variance. Some plates contained hundreds of colonies, while others contained none. And how they explain this is that there is some constant mutation rate, and therefore, if the mutation occurs in some early stage of the division, as you can see in the right panel of the right figure, then there are many tolerant cells and that are present on the Petri dishes. However, mutation may not occur at all, or may occur in the later periods of the cell division. So the conclusion is that the genetic mutation occur without the presence of external stimuli, and that is in favor with the Darwinian theory. And we will, our main focus is to look at the shape of the probability distribution of the resistant cells. So, as I said, the classical Luria-Delbrück model assumes irreversible resistance. That means that a sensitive mother cell, which is denoted in this figure as white ball, may have a tolerant daughter cell. Tolerant means mutant, which is denoted as black ball, with some probability denoted as mu. However, once the mother cell is tolerant, all other daughter cells will be tolerant as well. So that's the classical Luria-Delbrück test. However, we were motivated by recent research in drug resistance in cancer and microbial cells, which shows that these changes between the phenotypes may be just temporal, and therefore that cells may switch back and forth between the sensitive and tolerant state. We modeled it by introducing another probability denoted as lambda, which is the probability that the tolerant cell may have a sensitive daughter cell. And again, what we are looking at is the probability distribution of the number of resistant cells in the total population of size n. This probability satisfies this recursive equation you can see on the slide, and it is usually denoted as the master equation. And our main focus was to solve this equation or look for the approximations when the perturbation parameters lambda and mu were small. So our main results are as follows. First of all, in this presentation, let's assume just one initial condition, and that is that there is one sensitive cell in the beginning. All other cases are discussed in our article. And so the results are that when the perturbation parameters lambda and mu are small, there are basically three regions. On the left boundary, we have a left boundary solution when the number of resistance cells is small. On the right, the number of resistance cells is large, and in between them, there is the so-called regular coarse-grained solution, which is in fact the simplest, so I will start with this. We derived the formula as follows. It was derived by a regular perturbation analysis in the perturbation parameter mu, and we can see that it describes the bulk of the distribution very well. So if we compare the numerical results, the blue dashed, with this approximation, which is the red solid line, it works very well. On the left boundary, we derived by generic function and then some matching analysis this approximation where F Landau is the probability density function of the Landau distribution, which is one specific heavy-tailed stable distribution. And again, we can see that it matches very well. I have to mention that this approximation is not new. In fact, this holds also in the classical model where there is the irreversible transitions. However, what is new is this right boundary layer solution, which we found that it goes as the Landau cumulative distribution function. And again, you can see that the approximation matches the numerics very well. If we have the left, right, and the regular coarse grain, then we can develop the composite solution, which has this form. And again, we can see that over the whole range of M, it fits perfectly. And so we come to one of our main results, and that was to quantify the main effect of lambda. And that is that on the left and central part of the distribution, we saw that by introducing the reversible transition, mainly nothing serious happens, nothing serious changes. However, on the right side, a new boundary layer is introduced. As you can see in this picture, the black curve represents the classical model when lambda equals 0, and it ends with a sharp cutoff. However, when lambda is positive, the edge is smooth, and that's the boundary layer. And we know more. We know that it is described by the Landau CDF. So with this information, I would like to thank you very much for your attention. And if you have any questions, feel free to ask now or during the poster session. Thank you.